when we were talking about the starfish feeding on them, have in fact muscles, adductor muscles, that keep them closed. When the water comes in, they relax, the shell opens, and they filter feed the microorganisms out of the seawater. I've opened one here for us. In fact, I had to cut through this adductor muscle, which you see right here. I've cut through it with a knife to expose some of the internal structures. They hold onto the rock with these threads that are produced as a liquid by glands right near the mouth. When they extrude that liquid, it solidifies when it hits the seawater, and it forms a place of attachment for these muscles, firmly affixing them to the rocks. But if we look at their feeding mechanisms, it's kind of interesting. If I pick up this very thin filament layer right here and just touch it with the knife, it's about 2 cell layers thick, and if we looked at it with the microscope, you'd find that all the cells had cilia on them. They're ciliated cells. And what happens is when this mollusk opens up a little bit, when this water comes in, the seawater containing these members of the phylum Protista washes in, collects on the cilia, and the cilia act as kind of a bucket brigade, carrying all those microscopic food particles toward the mouth where they're ingested. So they're filter feeders. If we look down at the rock, we see some other organisms that we were familiar a little while ago. In fact, on the big rocks next to the shore, these organisms right here, which are gooseneck barnacles. Notice this gooseneck barnacle looks just like a shell here. But remember, when the tide comes in, the environment changes tremendously. When the environment changes, the water washes in, watch him open up. And there comes the organism out of his shell. He's lying on his back. He has these feather-like appendages. This is in the phylum Arthropoda, jointed-legged appendages. And they're filter feeders also, although they are in a different structural family, phylum. These are in the phylum Arthropoda and in the class Crustacea, whereas the mollusks and the mussels are in the phylum Mollusca. These also have bilateral symmetry. They're characterized by jointed appendages. Sometimes the jointed appendages can be seen very obviously. Other times they form mouth parts or antennae or tail parts. But the entire phylum is segmented. And each of the segments then is possible for them to specialize into different sorts of labor activities, such as feeding or crawling or hiding. Here's a representative of 2 different phyla. This is a snail shell, and inside this snail shell, phylum Mollusca, and inside the snail shell is a member of the phylum Arthropoda. It's a hermit crab. He's hiding in here. Let's see if I can put him in some water and trick him into coming out. I'll just drop him in this Ziploc bag. What they do as they grow, and they do get much larger than this, they leave the shell momentarily, and there's quite a competition set up for shells of different sizes among this group of organisms. Class Crustacea, phylum Arthropoda. This is the representative of the phylum Mollusca that you can see the bilateral symmetry. This is the black chitin. I said there were a variety down here. I just found the largest of the chitin that we have down here, the gumboot chitin. You see that it has the same general parts as the entire phylum Mollusca. It has the mantle and the foot, and the shells in this case are hidden in this fleshy back. Down in here are the gills. This particular animal, as well as the other chitins, are grazers. That is, they move along the rocks very much like a snail and graze the algae from the rocks. This is the lined chitin, one of the smaller varieties that we have. Okay, so far we've looked at a couple different aspects of the intertidal zonation. One, remember some of the physical causes for the zonation of the living organisms. Then there are the biological causes of predation and competition. Then we looked at some diversity. We found 4 different divisions of plants that are macroscopic plants, that is, the ones that are very obvious down here that we can see them. And we also looked at the phylum Protista, the organisms, the phytoplankton and the zooplankton that are microscopic, and they're floating around in the water. Now, for each of these organisms that I've covered, I'd like you to be able to kind of make a little mark on your intertidal diagram and then fill out that chart, organism by organism, and put each phylum in its proper place with some characteristics that are unique to each taxon. And the third thing that I wanted to talk about was some of the major feeding strategies, the dynamics of what's going on out here. We've already examined some organisms that I've called filter feeders. That is, they have appendages or gill segments that feed upon the organisms that are in the water itself, the phylum or the kingdom Protista. These are the filter feeders, such as the clams and the barnacles. Then there's another group of organisms that feed on its macroalgae, the reds, the browns, and the greens. Some of them are grazers, such as the chitons and the snails, that actually move over the surface of the plants and scrape material off. And then there are the shredders, such as some of the crabs, like that hermit crab that we saw, that actually take bites out of the algae. So we have 2 different aspects of the food web, the primary producers in the plankton and the primary producers that are macroscopic. And they're both fed upon animals adapted as herbivores. And the next thing in the food chain, of course, are the predators. And down here we have gulls in the background, we have cormorants that are fish eaters, and other organisms that feed upon both of these groups of organisms. If you do come over here in small groups, look underneath the algae or observe very carefully on some of the rocks, because some of these organisms, in fact most of them, have various strategies of hiding or collapsing when the water goes out. For instance, on this rock here, this looks just like a tarry material or part of the rock itself. But if you look very closely at these organisms in here, can you see that each of these is an individual sea anemone? And if I touch this group here, you can actually see this is living material. This is the colonial elegant anemone, or a little striped anemone, and each one of these groups here has been produced asexually, so all the members of each one of these colonies is genetically identical to every other member. In fact, they're clones. Here we find some red algae, sometimes called old man's beard because it looks kind of like a beard. Here's other members of the red algae, and barnacles that are filter feeders. And so now we've looked at the intertidal zonation, and as we pan up, we'll find that this is an example of where the zones are spread out and very broad because they're on a very level. And we've seen as that surface then gets more vertical, those zones collapse. But we find the same kinds of organisms and the same sorts of strategies of adaptation in each of these zones. Now we're in the upper intertidal, and we've also visited the lower intertidal, where are exposed only during the extreme low water spring tides.